participants who join us today. So probably we'll just get started. So hello everyone, good afternoon. I'm Cassie and I'm the moderator for this webinar today. Welcome to our webinar with the title One-Stop Solution for Library Design, Protein Engineering, Screening and Expression. We are thrilled to have you to join us today as we unlock the potential of protein discovery and target identification through an exclusive synergistic partnership between Allozymes and GenScript. Our speakers for today are Dr. Akbar Wahidi and Dr. Charlie Zhou, who are leaders in their respective fields. Dr. Wahidi, a scientist entrepreneur with a PhD in biocatalyst from the National University of Singapore, co-founded Allozymes to revolutionize enzyme engineering with his expertise in microfluidics-based enzyme engineering. He serves as a co-founder and chief technology officer of Allozymes, where he builds future-oriented solutions to accelerate enzyme engineering for diverse cell-free or cell-based applications in synthetic biology, chemicals, and cosmeceuticals. Dr. Zhou, with a PhD in microbiology and immunology from the University of Saskatchewan, Canada, is the head of project management department of molecular biology at GenScript. He leads the molecular biology project management team, providing support to molecular biology customers globally. So for today's webinar, we will cover the following topics today. We will explore the innovative microfluidics technology developed by Allozymes for rapid and efficient enzyme discovery and engineering. We will provide insights into GenScript extensive protein and target identification services. Then we will discover how this partnership between Allozymes and GenScript streamlines and accelerates the protein discovery process. And lastly, we will learn how this collaboration empowers researchers to unlock new possibilities in various fields, including drug discovery, biocatalysis, and synthetic biology. If you have any questions for our speaker, please leave your questions at the Q&A box and we will answer them after the presentation. So without further ado, let's embark on this journey of discovery together. So please join me in welcoming our first speaker, Dr. Akbar Wahidi. So Dr. Wahidi, the floor is yours.
Thank you, Casey, for kind introduction. I'm pleased to give a talk today with Dr. Charlie regarding our collaboration and our capabilities in one-stop solution, enzyme engineering, and library design and build and expression systems. Here is a very brief workflow how it works. Allozyme takes care of the AI and the data science part of it for designs. We design our libraries using cutting-edge technologies and methods, and we work with GenScript closely to synthesize those. GenScript has precision mutant library services, which accelerates significantly the quality and speed of our library builds. And then with our droplet technology that we have, it's our proprietary technology for screening. We have ultra high throughput screening capabilities, which allows us to screen up to 20 million enzyme variants after we receive such libraries, depending on the size of the library. And finally, this iteration may go a few rounds. And if we have a need for enzyme expression optimization and scale up, GenScript capabilities also comes in the expression systems. This is the overall workflow. Maybe next slide. Yeah, in here it shows a bit more detail and the benefits of working with us. We believe we provide in this partnership a greater purchasing power, meaning you save your time and cost of the project, especially protein campaigns. Allozyme can take care of even discovery all the way to designs of libraries and improvement targets to do a protein engineering campaign. And we work with GenScript as described for constructing such libraries. Allozyme screens, and this may iterate. But in this loop, we are providing 10x larger library, meaning because of the quality of the libraries that GenScript gives us and the variety of the library builds, we are having bigger diversity. And we are much faster, at least 60% faster in the overall screening flow. And we will describe more details of the screening itself. We are way faster than the other technology and bigger libraries we can screen. And the total campaign can take somewhere 2 to 4 months to build libraries, screen, and get to some improvement that is targeted. Here I will pass to Dr. Charlie to deep dive in how GenScript performs these steps.
All right, thank you, Dr. Bahidi, for the introduction and to the collaboration between GenScript and the Allozymes. So here I want to take on and expand on the content above to introduce our company and GenScript and the services we provide to our customers and partners in these collaborations. So basically, GenScript has been developing for more than 20 years since its establishment by our three co-founders in 2002 in New Jersey, the USA. So in the year of 2013, we established Baseline, and in 2014, we established Legend Bio, who are focusing on the industrial synthetic biology product and the CAR-T cell therapy, respectively. And in the year of 2015, we are listed on the main board of Hong Kong Stock Exchange. And 3 years later, we established the biologics business unit, which is now known as ProBio. And last year, our CRISPR gene editing GMP production facility was officially launched and put in operations. So this is the basic, our major milestone over the past 2 decades. And we also have a lot of detailed milestones, but in the case of the timing here, I'm not going to share details here. So could you please go to the next slide? Okay, I am from the Life Science Group in GenScript, providing the research services for customers from 4 major areas: and antibody therapeutics and vaccine development, and cell and gene therapy, and diagnostics, and agriculture and consumer goods. So we aim to provide reliable, high quality, and innovative reagents and instruments with superior customer service to enable success across a wide variety of existing and emerging life science research and development areas, or in a simple term, make research easy. Next, please. If we divide our CRO service according to the business scope, it breaks into 8 categories. So it includes the molecular biology services covering the DNA fragments, gene synthesis, plasmid constructions, and plasmid production, as well as mutagenesis service, including the DNA library. And we also have the gene editing service providing double-stranded DNA and single-stranded DNA synthesis, marker-free plasmid constructions, as well as a single-guide RNA synthesis up to the cGMP level. And for protein expression service, we are providing service based on 3 major platforms, which I will elaborate later in this talk. And we also have mRNA reagent services, which is one of the most popular services nowadays because of the success of mRNA vaccines. Aside from that, we also have a standard and modified oligonucleotide synthesis service, peptide synthesis service, virus and cell line service, as well as various reagent antibody production services. In the previous slides, I briefly introduced the business scope of GenScript in the CRO section. So next, I will focus on the DNA mutant library service, which is most closely associated with our collaboration with Allozymes. So the DNA library service of GenScript has been around for quite a long time, and starting from providing the degenerate mutant library to the trimer library suitable for the high throughput phage and yeast displays. But both types of library have various problems that are difficult to overcome, such as the homogeneity and the specificity of amino acids for the degenerate option, and the problem of being incapable of codon optimization for both types. So with the development of our high throughput chip oligo technology, the problem mentioned above have been solved. So in GenScript, chip oligo platform, we call it GenScript, has made a great leap in our library construction capability with the merit of elimination of unwanted codon or mutations, as well as a fully customized variance ratios and codon preference. Next, please. Our chip oligo platform is built on the semiconductor electrochemical synthesis method. Computer software activates specific microelectrodes by applying voltage to the chip, thereby controlling the chemical reaction on activated microelectrodes. Because each spot is independently controlled by the software, therefore theoretically each electrode can synthesize a unique primer sequence. With the precise control on each spot, we can determine the codon used for the specific amino acid and the frequency of each mutation. So currently, we are able to provide up to 8 million oligos at one time from one single chip. So the powerful chip oligo synthesis capability combined with 22 years of experience in gene synthesis and molecular cloning at GenScript, it really enables us to provide high-quality DNA libraries to our partners. The single sequencing, along with the NGS validation, help us to present the most accurate sequence and a uniform mutant distribution to our customers. And the comprehensive cloning experience we learned over the past 2 decades allows us to produce DNA libraries containing at least 90% of the theoretical diversity and a library size exceeding 10 to the power of 10. In addition, as a feature of all GenScript services, our DNA library service is also supported by experienced PhD-level field application scientists and project managers who are waiting to address technical challenges for our customers. So the content showing here on this page demonstrates the differences in quality between the precise mutant library constructed using the chip oligo and the traditional degenerate library. So the left chart shows the variant distribution in the library produced using both methods. We can see that the NNK degenerated method cannot introduce only the target variants at a specific site, and there's no uniform distribution at amino acid level. Under the same design, the precision mutant library not only accurately synthesized specified amino acid mutations, but also provided equal variant distribution. On the right part, the diagram explains the differences in variant coverage in 2 different libraries, achieved by picking individual clones after transformation. So it is clear that the precise mutant library can help customers get a higher coverage of total variants with fewer colonies picked in evenly distributed variant pool. It for sure benefits our customer on both reagent and labor costs and the screening turnaround time. So currently, we provide 3 major types of precision mutant libraries to our customers. A site saturation immunogenesis provides multiple sub-libraries. Each library contains all mutations from one single site, so making it convenient for customers to screen site-specific variants or to meet the experimental purpose of picking individual colonies or clones. So scanning saturation mutagenesis on the other side is more suitable for high-throughput screening methods. We collect all mutants and send them as one sample to our customers. And finally, the combinatorial mutagenesis will simultaneously introduce target mutation at all specified sites designed by the customers. And compared to the first 2 types, the combinatorial one usually generates a larger library size and contains more variants as expected. So, if everyone still remembers the collaborations map presented by Casey and by Dr. Vahidi at the beginning of this webinar, we could note that the GenScript service in this collaboration is not only limited in a DNA library, but also includes protein scale-up production. So I want to briefly introduce GenScript's protein service to you as well. So our protein service was launched soon after the establishment of GenScript with a total of 18 years of service experience. So till now, at least we have delivered at least 100,000 customer projects successfully to our customers, with a success rate of at least 98%. So GenScript protein expression service are provided based on 3 major platforms: bacteria, mainly E. coli, or insects, including SF9 or SF21, Pi5, etc., and a mammalian cell platform using both CHO and HEC293 cell lines. So each platform has unique service advantages, but most enzyme expression services are completed on the E. coli platform. The insect and mammalian platforms generally undertake more complex protein expression services. Next, please. So our E. coli protein service platform has the capability to provide protein service at various stages, from the early discovery with a high throughput manner to late stage commercial production. So offering high quality one-stop solution to our customers, especially for in the commercial GMP production stage, we can provide a maximum expression volume up to 20,000 liters. Next, please. Okay. So this is an overview of customized enzyme production we can provide to our clients using the E. coli platform. So for protein intended for commercial production, our process development includes cell banking, upstream and downstream process development, analytical method development, formulation development, stability assessment, and the final scale-up production. Okay, so I think that after knowing our capability of DNA mutant library and protein services, you must be curious about how a DNA library with such high diversity can be screened for target in such a short period of time. So in the next session, I would like to invite our collaborator, Dr. Akbar Vahidi from Allozymes, to introduce their company and its powerful technology.
Thank you, Dr. Charlie, for a great presentation. Yes, I will continue from here about how Allozymes accelerates and kind of complements what GenScript is providing. Here I will start with how we design and screen 20 million variants per day to help you succeed with our fastest enzyme engineering platform. Next, please. So very brief introduction about Allozymes history and milestone. I co-founded Allozymes in 2019 with my co-founder Peyman Salehian. Then we patented ultra high throughput screening technology in 2020, and we have raised our seed funding $5 million US to expand our operation and hire a bigger team to execute the plans. And also we won numerous awards as innovative biotech company in the region and beyond for the services and products we were working on. In 2022, we started officially our enzyme engineering and screening services, and we have served so far several industrial customers to improve their enzymes of need. And in that year, we also established our partnership with GenScript, seeing the great synergies we have. And the following year, in 2023, we recently raised our Series A 15 million US to expand our capabilities and team size. We established a new 10,000 square feet R&D lab in Singapore and expanding to Europe. And we have established also strain engineering capabilities on top of our protein engineering. Next, please. Here is a deep dive how we kind of function and what are the basically services you can expect from us in detail. We can carry from design to scale for protein engineering campaigns all the way to strain engineering. The core parts that we have is protein engineering or enzyme engineering, as well as strain engineering. In enzyme engineering, we often target properties of enzyme to be improved. That could be activity of the enzyme, stability of the enzyme, such as pH stability or thermal stability of that, and the selectivity, which could be different enantio, regio, or chemoselectivity. But often activity is one of the targets to improve the enzyme performance or reduce the cost of it in many processes. We also recently established our strain engineering capabilities to go beyond just a single enzyme and prove that our technology can work in different setups. So we have pathway assembly and optimization. Often we use E. coli and Saccharomyces cerevisiae for this part. We have metabolic engineering and we have strain optimization, and we have been working in internal projects and starting to work with external projects in this part. But in overall, to cover this enzyme engineering and strain engineering efforts, we have several buckets: library design, enzyme discovery, ultra-high throughput screening with our proprietary microfluidic technology, sequencing, and bioprocess. In the library design for the enzyme engineering, beside classical ways of looking at the structure and finding some potential spots to not miss, but we can go beyond that. New methods of such as machine learning methods of zero shot for single site libraries, or MLD, machine learning directed evolution for more combinatorial libraries we use in-house for designing our libraries, and we have our models trained for that. We build with these capabilities different single site, smarter or much larger, and we synthesize with GenScript. We also build our combinatorial libraries and screen very large diversities. For the strain, we have automated virtual assembly, which in short helps us to assemble in virtual settings and design and then just build it in the strain quickly. And we have genome scale metabolic modeling to target some very niche changes, knockout, knock-ins, and improve the strain. Also, we have genome annotation and primer design automated in our platform. In the enzyme discovery, beside the classical ways of screening large diversity of enzyme, we can also look for the databases and search sequences on similarities and diversities. And we can also look at the 3D prediction. Often we use that to make a smaller number of enzymes, and we synthesize the genes and we search for the target enzyme. We also have biosynthesis and retrosynthesis, which often comes helpful for the strain and pathway, because sometimes it helps us to have a better pathway optimization approaches. In the ultra-high throughput, which we will deep dive in coming slides, we can cover different strains, meaning bacteria and yeast. In bacteria, E. coli and Bacillus, we have worked extensively for expressing enzymes and screening them, the variants of enzyme. In yeast, we have Pichia and Saccharomyces cerevisiae extensively used for our project. And then we also have covered various classes of enzyme. To date, we have covered 2/3 of main enzyme classes that are out there, and we are expanding on that. And expression systems, we are very versatile. Our droplet system works as classic microfluidic system. We can work with whole cell system, which is intracellular expression, or intracellular lysel systems. We can also work with extracellular expression systems, as well as cell-free expression system. All of those are compatible with our platform, and we have used them. In the sequencing, to accelerate all this screening, because we have very rapid screening, but we want to also identify the hits and next steps, we have extensive in-house DNA sequencing capabilities for various types of samples and fragments. We also have long read and short read pipelines in-house, and we can also do whole genome sequencing. Often for bacteria, we run that. For bioprocess, this is to build a baseline bioprocess, because it's often not easy to just have some shake flash data and just convert it easily. It takes longer, but we have our in-house capabilities to do a baseline bioprocess optimization, and up to 150 liter nowadays we can run in-house for our strains that I mentioned we are working on, or if we have any interesting target in collaboration with a partner like GenScript, which they can go beyond that, and they can go thousands of liters even if needed. And we have also labs scale DSP and quality control capacities. This helps us to accelerate initial sample preps as well for the enzyme of interest or the target compound. So we have these core services and smaller services. In the next slide, please. Yes, in short, current enzyme engineering is sophisticated. In the given timeline and budget, it's often difficult to hit the target. That's why we are calling here failure rate is high. Doesn't mean there's no improvement at all, but it doesn't hit the targets often. And scalability, you have to deal with hundreds of samples per day, and whereas normally you have to screen thousands and millions in this field, that's not an easy job to do with the current technology. And the cost of it, of course, each enzyme and gene campaign can go to be beyond $1 million and above that, definitely in many campaigns and reported papers, if you have a look at the rounds of evolution they have conducted. And that makes also a slow process overall. And let's not forget, often you don't have one enzyme, you may have more than one enzyme, and this adds up to the issue. Next slide, please. So what we do is we are having our proprietary droplet microfluidic technology that miniaturize and automates the current technology, meaning we don't use microplate for screening. And this allows us much faster and larger libraries to be screened. The speed of it and the size of it change significantly. That means we not only we can do the current projects better, but we can also do things that are not possible before, or the enzymes that needs much larger library to be screened by nature. In the screening itself, meaning when you just have the screening of the variants, when you have a large combinatorial, a very large, diverse single-site library, we are 2,000 times faster. No screening goes beyond a day almost for us, whereas the classic ways of fraction of library takes few weeks. Cost efficiency of the consumable, we are significantly better, given that we screen much larger number of samples, we use much less reagents, as well as many consumables we don't use a number of tips or plates. And we are more reliable in a sense because we cover larger libraries. For a given library that you have to just take a fraction randomly and screen that, we cover it and we give you much more reliable data, kind of not missing the best hits out there. And we are more scalable number of variants in each one we can screen. Next slide, please. Yes, and here we will show you a short video. How do we screen this 20 million variants a day? Just some simplified way of doing that. It's a video. We will play it now. Yes, so in classic way, instead of thousands, we can do millions of variants built and encapsulate after expressing them in the cell. Each cell has one enzyme variant imagined, and then they can be also incubated over the time, and you may have some of them performing better than others. It goes through our proprietary microfluidic system. Using our hardware and software, we can sort them. The best one goes to our collection, and the rest will be wasted. We can also, this shows we can accelerate this instead of weeks to days or hours, the screening, and that saves a lot of time and money for our clients and opens new capabilities. Next slide. So we have prepared one case study that I will explain a little bit how that project was done, and Dr. Charlie will also kindly show how that library was built and the quality of that. So this case study we have prepared is a transferase, which is a transaminase indeed. It's a very popular enzyme in industry. Many examples of that has been used in the industry. We improved this enzyme 10 times in 2 months, which I'm going to elaborate. The partner that we work for spent nearly 3 years, and they had very limited success. To be fair, they work with us to outsource and help to accelerate because they had very tight deadlines that year coming. So we took that project. In a week, we designed the library. GenScript helped to accelerate the build of that library in less than a month for us. And then we screened a single site 7,000 variants, which was designed. And we quickly found 5 hotspots. We combined that. We built a fully saturated simultaneous for the 5 spot, which makes 3.2 million of variants when we saturate all the spots at the same time. And we screened it in our platform again, and we found final 10 times improvement over activity with a rare mutant in 2 months. And this was interesting because we also run some plate screenings for some of these samples, and we never managed to find those, I would say, rare variants that we found in the plate screening. It's just because you don't really screen those large library in plate, and we run some tens of plates over the time. We never encountered those variants in the plate, whereas in microfluidics we found some very rare and very improved variants. This was showing the speed and the power of our technology. Next slide, Dr. Charlie will show how they synthesize such libraries.
Thank you, Dr. Vahidi. So for this case, once we confirmed the library design, Allozymes, the production got kicked off right away. So eventually we managed to deliver a 500 nanogram of double-stranded DNA library to Allozymes within 1 month. So the NGS was able to identify 99.94% of the target variants from the final library product, and the mutation at each site were eventually evenly distributed with little fluctuation, as I shown on the left part as an example. So we further amplified the Sanger sequencing on random colonies to determine the positive rate of the library, and the result shows that 80% of tested clones were correct. Dr. Vahidi.
Yeah, thank you. Yeah, so here, if we want to compare 3.2 million variant screening, what happens? We were much more cost effective in that process, and we were 2,000 times faster in the screening itself. Whereas if you wanted to screen that with automated microtiter plate, not the manual ones, it may take up to 100 months beside the huge cost of it. But okay, what happens is in reality, we can still screen that in a matter of a day, and we can do the follow-ups, whereas in the current technology, in the plate-based automated systems, they would just go for a fraction of that library. Often a percent or less than that will be just picked, and the colonies will be screened, and a few hundred up to 1,000 at best. So the best rates that we know in the industry that can cover just to up to a few thousand samples. So there's 3 order of magnitude difference in the throughput here in whatever way you want to calculate. So that means that the compromise is you go with much smaller library size, and this often makes a difference because most of enzymes, I would say, are unknown and not really studied, and you really need to screen larger libraries and find the combinations and the spots. When you want to be so smart in every design, that doesn't pay back easily, and that's why you have very huge number of rounds of screenings or failed projects. Just the improvement doesn't make enough sense. Whereas Allozyme technology helps to support that part without really adding up to the cost much or time much. And next slide, please. Yes, so we are more than happy to discuss your projects ourselves. Dr. Deepak, our business developer in Singapore and Asia Pacific, he has a PhD in biotech as well. Dr. Audrey, same, she was scientist and worked in the top-notch companies and enzyme Excodexes, and she's in Europe based, so different, and US she's covering as well. So you can directly connect them and explain if you have a project in mind or if you want to learn more about us. They are more than happy to guide you. Definitely Dr. Ethan Lee, also from GenScript, that they do have their own customers and services that you are interested in. And definitely, if you want to learn how through GenScript, you can connect to us and benefit from this partnership. Dr. Ethan is more than happy to explain further about how we are working, what is the model about that. But it's very simple. You really don't need to do much. It's just more like describing your problem and we coming with a proposal to that. So with that, I would thank everyone for attending this, and I believe our seminar, that talk is finished, and we are more than happy to see any Q&A come.
Yes. Thank you, Dr. Joe and Dr. Vahidi, for the great presentation. So for our attendees who are online right now, please feel free to drop in your questions if you have any in the Q&A box, and we will answer them right now. So we have a few questions coming in already, so we'll attend to them one by one. So the first question from Raja. So he's asking, what are the key factors to keep in mind when synthesizing a gene of interest, whether it belongs to bacteria or fungi? Also, what can be done if the synthesized gene fails to express in a host? Additionally, is the synthesized genes given separately or with a vector? I believe this is for Dr. Joe to answer.
All right, I'll take it. Thank you for asking this question, Rajan. And you have three sub-questions. I'll try to answer one by one. So the key factors when doing the gene synthesis is the sequence accuracy, I think. So whenever you're delivering the fragments to customers or clone that into a vector and deliver as the clonal genes, I think the accuracy of the sequence that's synthesized by us or by other vendors is the most important things. But if you have troubles when you have this like plasmids or gene of interest that fail to express in the host, I think it comes to a little bit more complicated cases where you have to really analyze a lot of things. I think, for example, you have to really double check whether the vector you're using is correct for the systems or the expression host or for expression cassette from the promoter to the terminators, if there is, as proper for this host. And in some cases, we have customers approach us, but using either a wrong selection markers or the concentration of the marker is not correct for the use. So it could be one of the cases. And also, I have to bring into cases that sometimes the gene of interest will somehow interfere with the host. In most cases, it's E. coli. So the potential gene toxicity is another issue that you have to double check before you really do the expression. Whether overexpression will cause any issues for the to the host, then you have to try to manipulate to the expression level, trying to lower down the toxicity brought by the overexpression of the gene. And so I think that's one of the things. But I mean, like it could most of the cases will happen when our customers really, either order a plasmid from us or they order fragments and clone it in-house to target vectors. So I think it should come from the vector will be most cases, but just keep in mind that if you have fragment, unless you're using a cell-free system, you somehow will come to these cases, will come to this situation.
Thanks, Dr. Joe. Another follow-up questions from Raja again. So what are the costs for the services for gene synthesis and mutant library construction and screening?
All right, so for gene synthesis, I think in GenScript, we have a different levels of synthesis with like a different needs for some customers who are ordering or in needs of like fast synthesis. We have a rocket synthesis service which can provide you with the turnaround time as short as like 3 business days for production. And of course, the turnaround, the price will be a little bit higher, but for some cases, if you're in tight budget, but you really need the plasmids, we also have economic service that will be suitable for you with affordable prices. But the turnaround will be a little bit longer, but not that long as like accounted by the month. So we can talk to you in details about the needs later by our colleagues or by myself as well in terms of how we can help you with it. And same for our library service as well. So for library service, we have 3 major types, and also for deliverables, we can deliver either the double-stranded DNA as a fragment, or we can clone the fragments into the vector of your choice. And for sure, that will be additional fee for the cloning, but I think a lot of benefits is that we have a rich experience in terms of the clonings. We'll keep their high diversities and the coverage as well as the quality of the libraries for you. But you're really in like a tight budget as well. You can try to try our double-strand DNA libraries as well as a starting point.
All right, thanks, Dr. Zhou. The next question from Nava. So in the design process of the variant libraries, is structural information taken into account?
Yeah, I will take this question,
sorry.
I believe.
Sure.
I believe it's a variant, enzyme variant library question. Yes, we do look at that. In fact, in our design concept, we take more than 5 approaches to make sure the design comes into consideration of all the important factors, structural factors, order factors, and more, as I explained, newer models that we are using to not miss any good spots. But let's not forget that we are not heavily depending on that. If the enzyme is really not known or not well kind of studied, we can go larger libraries of aggressively mutating the enzyme across the sites, and with GenScript power of synthesizing that, we can have very large diversity as well. But in terms of being smart and design, yes, we do have those, including the structure-based spot selection.
Thanks, Dr. Vahidi. The next question from Jiran. Do all the droplets contain enzyme variant or majority of the droplets are empty? And what is the rate of false positive from microfluidic screening? I believe this is for you to take.
Yeah. Yeah, that's a good question. So this is called the encapsulation rate. So I cannot disclose exact encapsulation rate, but I can say yes, not all the droplets contains the basically samples. And the 20 million sample we are saying is not 20 million droplets. We go through much more than that in a day, and that's really calculated based on the encapsulation rate. When we are saying 20 million samples, meaning 20 million cell kind of the droplets that contain samples. And in terms of the false positive, we are very accurate. The system we have is 99.8% accurate, and we have different QCs and a sophisticated hardware that helps us during the sort, make sure the sort goes well, or if not, we will pause and repeat that, which is one of the benefits of working with us, because we don't need to run a few weeks and learn something. We can just quickly iterate and improve the process without really costing anything to customers. So, but we have by nature 99.8% accuracy in our system selection.
Thanks, Dr. Vahidi. The next question from Yi Fang. For the library design, how do you decide which amino acids to be mutated? Is it based on the known or published work database or something else?
Yeah. Again, here I can deep dive a bit. If the enzyme is known or published, we take advantage of those publications, especially the structure of it or any other things, then we will benefit. Of course, now there's other softwares like AlphaFold also helping to know the structure, but sometimes it may not predict well for certain very rare enzymes, I would say. Overall, if the enzyme is known and you have good information about its mechanism and other things, yes, we do try to be smaller size in the single site library. It doesn't mean just a handful. We go much bigger than always, bigger than that, because often you will find variants that are distant from the active site or other sites that you expecting it. But as I mentioned, in worst case scenario, imagine you have no clue about this enzyme, we can screen the full coverage of that gene, and GenScript can synthesize, and we can screen without any much issue. But that cost adds a bit of the cost of library, basically, and a little bit of the screen, but not too much, but it can be done. So that's not I would say, a challenge that you cannot tackle.
Thanks, Dr. Vahidi.
Just final to that, after we find the spots, we often come up with this scanning library or single site library screening results. We find the hotspots, meaning those are showing improvement, depends, it can be percentage or folds. And then we combine them. That's again our power, and we use our methods of design, which ones to combine together if there's numerous sites to combine. And then we build libraries and we screen them in very large numbers, because combinatorial libraries often get very large when you are running saturation libraries. And there we can find the best combination, because not the original amino acid that you find as a hotspot will be the best when you combine with the others.
Thanks, Dr. Vahidi. So the next question from Benny. What is the role of AI in library design? Can you briefly elaborate on that, please?
So AI is emerging technology here, and we are starting to use it. And I kind of mentioned here and there in library design explanation that we are not just depending on this design concept, but we are leveraging on those. And we have started this since last year. And AI, meaning we are using the models to understand basically which positions are potential hotspots. It gives us a good head start. Sometimes it helps us to reduce the size of library or the cost of build of that. And also from every screening that you do, we train our model with that. We say, okay, these spots are coming or these combinations are coming. And then these models are learning more and more. So AI is more or less for us works like, I'm talking about large language models. Of course, I'm not expert in the data science, but I know very well, like you have supervised or unsupervised, but whatever model is, we are training them and retraining them and adding more and more enzymes and classes of enzymes, and they are getting smarter. So the idea is them helping us, these models helping us without having specific pre-knowledge of that enzyme, to be smarter in the design of that library and be faster or cheaper the synthesis of that. So these are the targets, but AI, I would say in our field, is still in the early stage of that. It's not like you can have some AI models and you can solve all your problems. That's why I believe approach like Allozyme taking, that using those technologies as well as leveraging heavily on the screening and the wet lab capacities, that is the way to go for next, maybe long time, next few years at least.
All right, thanks, Dr. Vahidi. Next question from Joshua. Some industrial enzymes requires a very high titer and production. So can GenScript support Aspergillus? If not Aspergillus, is PCR better than E. coli? I believe this is Dr. Joe to answer.
All right. Okay. So short answer for the first question is that unfortunately, no, we don't have a platform for Aspergillus enzyme production, so using this as a host. But we do have E. coli, as I mentioned, and we do have yeast platform as well. But I think in comparison between I think E. coli and Pichia expression platform, we are more developed in the E. coli one with much more technology involved and developed there. So in terms of the fermentation yield, I think yield is for sure outpass the Pichia platforms. So I would suggest to really start to use the E. coli platform to do the test and to see if when we do the scale-up production, we'll have higher titer.
Thanks, Dr. Zhou. Next question from Yifang Yin. Can you introduce a little bit about using directed evolution, especially MLD you mentioned in the slides? How does it work?
Yeah, so in very simple way, direct evolution is on the iterations we make, right? So often we find spots, we combine them. Every time you find a spot and you make sure of that performance, you can incorporate that. So when you're incorporating your best variant and rebuilding the library or the new variations, you can you are just kind of practicing directed evolution concept. That's how direct evolution works. Some winner comes, it stays in the target, and then some mutation again comes to that, and you find other winners and other mutations. So that iteration is direct evolution to me. And MLD helps to learn with lesser data over the time. It's not like you can just jump to it and it will give you final data. But if you screen and really fit this machine learning direct evolution model, that then it will reduce the kind of size of library you need over the rounds of evolution. And this iteration for us, we can go 3 to 5 round of evolution often to hit target, but I believe in classic ways of play, there are a lot of reports more than 10 rounds, 15 rounds of screening. So this is the how direct evolution works with us and the kind of the rounds of it.
Thanks, Dr. Vahidi. Next question from Harry. Could we use droplet reaction system for single cell sequencing?
Short answer is no. So because your droplet reagents and composition is quite complicated and is not like built for sequencing. Often those single cell sequencing droplets or reagents are just built to just do a follow-up of sequencing, whereas we have to do the reaction first and find the winners. So we do take a different approach on that. Yeah.
Okay, thank you so much. So this question is directed to Dr. Vahidi from Li Liang. She has two questions. The first one, it seems that the construction of mutation libraries still have certain randomness and blindness, so it needs to have a large library to screen. Is there any way to make the process more targeted? Next question is as to the high throughput screening method, what is the limitation of MFC-based method?
Sure. So answer to first question is, as I mentioned, unless the enzyme is very well established and kind of reported, which very rare, very few enzymes are like that, and certain customers only have that capacities for very niche enzyme they work with. There is always this randomness. That's why like it's really like a problem of looking for a needle in a haystack. Enzyme engineering often is that the case. And just to get improvement on that, as I said, you cannot get to the best design in the one shot. That normally doesn't work for most of the enzyme engineering campaigns. We are using, I mentioned, newer technology to reduce the size of library and the efforts, but it doesn't eliminate or makes it very small library often. So I believe machine learning, AI, and better structure now with AlphaFold and others will help to be more targeted, but it will not eliminate the need for screening. That will stay there for enzyme engineering campaigns. And question two, MFC, I'm not really sure stands for what, but oh, microfluidic, maybe? Is it microfluidic? So if you can clarify, I can answer to that. I'm not sure what is MFC.
Sure. I think we can clarify with Lian later by email.
Yes, thank
Yeah,
you.
can. So moving on to the next question from Kavia. She's working on the COMT genes in humans, which is subjected to 6 exons. Is it necessary to design a primer for an intron to those sites? If not, mutated from an exon site? What's your suggestion on this?
Is this me?
I believe so.
All right.
It's something to do with
Okay. All right. I think for the expression purpose, I think if we're doing protein engineering, I think we should target on cDNA level. So in that case, I think mutation should be mainly focused on the exons instead of the introns. So I would suggest you to start with the exons first and to test out all the variants possibilities, and then if nothing really happens, and I will see if there's any issues or any possibilities we can dig in more into the intron.
Thanks, Dr. Joe. Next, we have a question from Vasilka. Do you set up the screening process for each enzyme separately, considering that they have different enzyme activities?
Yeah, so I would say we have this feasibility kind of in the beginning of project. We check the feasibility of that project. Can it be quickly run through our platform? So if that goes, we call that step a validation step, which takes answers to some of the follow-up questions as well in a way. It takes a few weeks only to validate and confirm the assay and then go for screening. But let's not forget, if there's such step needed, but we gain huge speed afterwards. So that is not really a bottleneck of this technology. It's just the need to make sure. And I think it's not exactly comparable, but in other HPLC or other methods also, you often need some establishment and development of method and validation as well. This is not unique for us, but this may take different steps to validate. It's very similar. So yes, in each enzyme process, meaning we have to validate and make sure it goes through. So we will go step by step, and that's how for special service projects we have step-by-step go-no-goes, and that each step is taking care of that. And then for the time I mentioned, it takes a few weeks of the assay, and we screen both existing assays we have, which we have a really good range of assays we have used so far, and we also can couple new assays for the existing assays to new potential targets, and that we have done in recent projects. So that's not an issue. So we can expand if we believe there's a value, and depending on the timeline of the project. If something needs to be immediately started, yes, we will be more comfortable with the existing assays to start with. And in terms of substrates readout, we are not limited to fluorescence, but we highly prefer fluorescence because of the throughput it gives. Absorbance can be used, and mass can be used. We haven't used mass so far, but we have some future plans for that. But none of those have the throughput of the fluorescence. And we have very good expertise on getting fluorescent readouts from assays without disturbing. So we call those label-free assays we build.
Thanks, Dr. Wahidi. I think you answered most of the questions. I think Liao just put in her clarification. Yes, she does means that is the microfluidic based screening method.
Oh yeah, yeah, it can be used for different proteins, depends on the properties. So I can just mention that you should just imagine our droplet system works like well plate system. It can hold large or small cells. It can cover different properties of the enzyme, different expression system. As I mentioned, even different pH or temperature, it can be very tolerant to the. So we are just mimicking what is done in the plate into droplet. So instead of well, we have this droplet as a vessel, and that accelerates and changes the throughput significantly. It doesn't limit as such. Very few other small limitations. Yeah.
Thanks, Dr. Wahidi. That's quite a number of questions, and we have successfully answered them all. So good job, everyone. But for those attendees who still have any questions after the webinar, right, just keep sending to us and we will answer them via email later. So as we come to the end of this webinar, I wanted to express my heartfelt gratitude to everyone for joining us today. It's been truly an insightful and engaging experience, and I hope you have gained valuable knowledge and inspiration from our distinguished speakers today. So I would like to let everyone know that today's webinar has been recorded, and the recording will be made available to all of you in the coming days. So this will be an excellent resource for you to revisit the content and share it with your colleagues who couldn't attend the webinar today. So shortly after this webinar, you will receive a survey form, and I will kindly request you to take a few moments to provide your thoughts and suggestions, because your input will guide us in delivering even more informative and engaging webinars in the future. So thank you, Dr. Cho and Dr. Wahidi, for the great presentation, and also to everyone who take the time to join our webinar today. And hopefully we'll see you again in our next webinar. So thank you and goodbye, everyone.
Thank you. Goodbye.
Thank you.
Bye-bye.
Bye-bye. Have a good day.